Welcome to this session of the Global Hog Industry Virtual Conference. I'm Andy Vance, host of the Feedstuffs in Focus podcast, and I'll be your moderator for today's session sponsored by Natural Biologics. Our topic is using membrane biophysics to beat African swine fever and other tough disease challenges. In this session, we'll take a look at ways we can target some of today's most pressing viral disease challenges at the membrane level, both in feed and in the animal. Our speaker, Dr. Charlie Elrod, will explain the mechanics of membrane biophysics and the practical implications of formulating precise combinations of monoglycerides, surfactants, and immune-supporting oligosaccharides to inactivate lipid-enveloped viruses like African swine fever while supporting the animal's immune system. Now, Dr. Elrod's been involved in the dairy industry for 40 years, and after building and operating his own dairy in the 80s, he went to work with the dairy and beef herds at Berry College while completing his Bachelor of Science degree. Then moving north to Cornell University, he completed his doctoral studies focusing on the intersection of protein nutrition and fertility in dairy cattle. Now, after completing his PhD, he spent a decade as a member of Cornell's University Extension faculty and directing the New York State Cattle Health Assurance Program. Over the past 20 years, he's worked as a nutrition consultant, tech service consultant, and leading R&D for the dairy division of an animal health company. Currently, he's the president of Natural Biologics, a product development and innovation company focused on helping solve animal health challenges. Just in 2018, he was appointed an adjunct assistant professor in the animal science department at Cornell. Dr. Elrod, the floor is all yours.
Well, thank you, Andy, for the introduction, and I'm really pleased to be here today. My first presentation to the Global Hog Industry Virtual Conference, but I think we've got some really cool data to share with you, and so again, I'm very excited to be here, and thanks for the opportunity. I'd like to start by acknowledging my collaborator, Dr. Joshua Jackman from Sungkyunkwan University in Korea. He's the real membrane biophysicist and has really helped us understand a lot more about some of the compounds we'll talk about, as well as some of the pathogens and challenges that we're trying to overcome. So I don't need to tell anyone in this audience that African swine fever virus is likely the biggest health challenge that we're facing in the global swine herd today. But of course, for several decades, we've been facing other challenges from lipid-enveloped viruses like PED and PRRS. And while there have been a lot of natural compounds introduced into the livestock industries over the years, a lot of times they've been fairly poorly defined and maybe not backed by the best science in the world. So what our approach has always been now to really understand the fundamental chemistry of both these compounds, as well as any target organisms that we're looking at. And we believe that that can really assist us in focusing our selection of bioactive compounds to reduce the specific health challenges that animals are facing. So quickly, I'm going to provide a little bit of overview of the medium chain fatty acids. There's been a lot in the press about them in recent years, and but a little bit of understanding of their chemistry, I think, is warranted. Now, this table is adapted from a review article which we published earlier this year on medium chain fatty acids and monoglycerides as antivirals and antimicrobial agents. So basically, you're all familiar with the structure of these medium chain fatty acids. They are 6 carbon saturated, well, 6 to 12 carbon saturated fatty acid chains with a carboxyl head group. And I'll just highlight that for a second. They have, obviously, as your chain length increases, increasing molecular weight, you'll see that the shorter the chain length, the lower the melting point. So some of these would be liquid at room temperature. But I'll also draw your attention to the column listed CMC, and that is the critical micelle concentration. And we're going to be talking about that some. So you can see for these free fatty acids, what we would consider to be relatively high critical micelle concentrations, 3,500 micromolar for capric acid or 900 micromolar for lauric acid. I would also draw your attention to the last column on basically the odor of these compounds. And again, with the shorter medium chain fatty acids, the caproic acid, for instance, C6, has a very strong pungent odor. And so could, if using that in a feed, could put pigs off of the feed. But as the chain length gets longer, basically the odor reduces with that increasing chain length. Now, as we move over to the monoglycerides, and we're calling these MCMG, or medium chain monoglycerides, we basically have the exact same saturated carbon chains, but instead of that carboxyl group, we have a glycerol attached to the head group of this monoglyceride. And so there's just one fatty acid attached to the one glycerol, generally and most effectively in the alpha or number 1 carbon of the glycerol molecule. These have higher molecular weights, higher melting points, and very importantly, they have much lower critical micelle concentration. So you can see for the monocaprin, about 600 micromolar. Compare that to the lauric acid, which was 900 micromolar. And then for monolaurin, which I'll refer to for the rest of this presentation as GML, glycerol monolaurate, has a very low critical micelle concentration of only 60 micromolar. And you'll see down the last column that all of these monoglycerides have very low odor, just minor smell to them, so there should be no issues with stability, palatability, or anything when we add these to feed. So again, I want to dig into this critical micelle concentration concept a little bit because it's really quite important. So they call these class of compounds that have, say, a hydrophilic head and a hydrophobic tail, they call them amphiphilic compounds. So what these will do is, in solution, basically the hydrophilic head will form to the outside and the hydrophobic tail will form to the inside of a micelle. But they won't do that until you reach this critical micelle concentration. So at and above that concentration, you will see the formation of these micelles. The interesting thing as we talk about these membrane biophysics is that free medium chain fatty acids or monoglycerides will not interact with bacterial or viral membranes. They have to be in their micellar form in order to interact with those membranes. So this is some data from Dr. Jackman's lab, a recent publication of theirs, where they actually looked at different combinations or proportions of lauric acid and glycerol monolaurate, or GML, and they can basically tune the proportion of that fatty acid with that monoglyceride and study their interactions with different membranes. But in these lipid-enveloped viruses, they will rupture and leave large holes in the viral membrane where basically either the DNA can leak right out. In the case of, say, a PED or PRRS, it has a very simple membrane, or it can prevent fusion of that membrane with a host cell and thereby knock down the virus's ability to infect or replicate inside a host cell. So let's jump into the data because this is the exciting part. I'm going to show you some data from a paper which we have just gotten accepted into the Journal of Animal Science and Biotechnology, where we worked with the National Academy of Science in Armenia, where they've been dealing with the African Swine Fever virus for a good 10 years or more. So this antiviral defense group there, we collaborated with them to conduct a number of studies with ASF. Now, I'm going to show you some data that has both virucidal or antiviral activity, and it's an important distinction. The first couple of slides, I'll show you virucidal activity of the medium chain fatty acids and GML on the African swine fever virus. Now, in these virucidal assays, we incubated the virus with the different compounds at a 5 millimolar concentration for just 1 hour. And then we diluted those suspensions basically so that when we took it into the next steps of viral infectivity, the test compound, the fatty acid or the monoglyceride, was at such a low level as to not for any way impact that infectious process. So this would be just looking at what does this compound do to the virus within that first hour of interaction, okay? And so what we can see here is that all of the medium chain fatty acids, so from caprylic, C8, to capric, C10, lauric acid, C12, and then the glycerol monolaurate, they all led to significant reductions in infectious African swine fever virus. The capric and the GML led to a greater inhibition of the infective virus. So about a 1.7 log reduction, which is, sometimes people say, oh, 3 log, 4 log reduction. A 1.7 log reduction equates to about a 98% reduction in infective virus. So a really significant reduction in the amount of infective virus here. Now, in the next series of studies, we wanted to take the concentrations down even lower. So we took it down to 250 micromolar. So this is 20 times less of the test compound in the incubation just for 1 hour. And then it was diluted, as in the previous assay, and added to these cells. We incubate those cells for 3 to 4 days until the negative control basically gets completely overrun by the virus and dies. So at that point, they take it out and they do a number of dilutions and they can determine the viral titer. So again, even at this very low concentration, 250 micromolar, the GML resulted in a 1.7 log reduction in infectious ASF. So very similar to the 5 millimolar results. Again, about a 98% reduction in infective ASF. The medium chain fatty acids at this concentration had no effect on the amount of infective virus. Now, in the next series of assays, we're going to look at the antiviral effects of the medium chain fatty acids and GML. So the distinction here between the antiviral and the virucidal is that the test compounds were left in this virus solution as it was plated into these viro cells to look for infectious properties. And so what that does is if the compounds interfere with some other aspect of either viral fusion with the host cell or entry into the host cell, perhaps unpackaging the viral membrane or even repackaging or budding from the host cell. It could be any of those steps at which these compounds interfere. So this would be a combination of both the virucidal and any other effects it may have on virus infectivity. So again, very similar setup, just whether we've diluted that initial virus suspension to reduce the level of test compound. And in this run at the 250 micromolar, again, you can see that the medium chain fatty acids had no activity on the ASF virus, but at this very low level, 250 micromolar, the GML had additional antiviral activity. So it took it from a 1.7 log reduction to a 2.8 log reduction in infective virus in this assay. So then we really wanted to see just, how low could we go with the GML and still maintain that antiviral activity. So we tested a number of concentrations from 16 micromolar up to the 250 micromolar. And what you can see is that just as we get above GML's critical micelle concentration, we start to see about a, we'll call it a 0.8 log reduction. As we get to 125 micromolar, we see about a 2 log reduction, and then 2.8 log reduction as we get to that higher concentration. So again, we're having both virucidal activity as well as disrupting some other step in the process of viral infection of these cells. So similarly, we've also conducted some work with Dr. Diego Deal at the Cornell University College of Vet Medicine. He heads up the virology group in the diagnostic lab. And we ran this test to look at different monoglycerides and lauric acid interfering with the PRRS virus. So basically, we use the higher concentrations, 5 millimolar of monocaprin, monocaprolin, lauric acid, and GML, incubating with the PRRS virus for 1 hour. These were then added to cultured Mark 145 cells, and after 3 days, the virus titer was read. And what you can see is that the monocaprin, it led to about a 1 log reduction in the amount of infectious PRRS virus, but all of the rest just completely obliterated the PRRS virus. So, highly effective against this virus. Very similarly, we just recently tested the same compounds, same setup. This assay is created using Vero cells. Not that that really matters to anybody out there, but you can see a very different result, that they all resulted in interference or antiviral activity with the PED virus. But again, GML just completely obliterated the virus and reduced its infectivity to essentially nothing. So, all of the other monoglycerides and even lauric acid were quite effective, but GML still proved to have the highest level of activity here. So I guess the point of some of this is that the phospholipid membranes of these pathogens are different depending on this particular pathogen, whether that's a viral or bacterial pathogen. So for instance, gram-positive bacteria have very simple single-layer membranes, whereas the gram-negative bacteria tend to have more complex inner and outer lipid membranes. Similarly with PED, a very simple single-layer envelope. And then ASF has the 2 lipid membranes sandwiching a protein capsid. And we're going to talk quite a bit more about that protein capsid here in a couple of minutes. So really, the efficacy of different fatty acids or monoglycerides will depend on the membrane composition of the targeted pathogen. And so we can tune our approach depending on the pathogen of interest, or we can, add enough in there to provide a broader spectrum of activity against a broad spectrum of viruses. I'll just touch briefly one slide on another aspect of lauric acid or medium chain fatty acid or monoglyceride activity, and that's anti-inflammatory activity. Well, we wanted to look at some of the other forms of lauric acid, so the free fatty acid, also a methyl ester of lauric acid, and then GML. And what you can see here is basically in a very dose-dependent manner, all of these different forms of lauric acid had significant anti-inflammatory activity. We did this work with Barry Bradford's group at Kansas State University, and it's really quite interesting, and another reason we want to build these into our end products, because they can help reducing that inflammation, helps blunt the effects of a potential infection. Another aspect, and actually a very positive attribute of using these fatty acids or monoglycerides, is their effects on growth promotion. So in this review we recently published, we reviewed a whole lot of the literature relating to various combinations of fatty acids and monoglycerides sometimes by themselves, sometimes in blends, and sometimes incorporated with other things like direct-fed microbials, prebiotics, short-chain organic acids, whatever it might be. But I would say the overarching theme of this growth-promoting activity were improvements in nutrient digestibility, improvements in the microbiome, shifting away from pathogenic species and more towards beneficial bacterial species, and then reductions in gut inflammation and permeability, with, of course, the things that go along with that, improvements in the structure of the intestinal epithelium. So, higher crypt depth and all those good things. So again, growth promotion through better digestibility, a healthier gut, getting more of those nutrients into the animal to do with what she needs to do. So these generally lead to improved average daily gain and feed conversion ratio, while actually performing similarly to any antibiotic treatments. So, a very beneficial reason also to have these included in your swine diets. We've also actually have borrowed some data from our friends at Berg and Schmidt who sponsored this study, and I'm sure you all are familiar with the Pipestone Challenge testing for some of these viral mitigants, where they take an ice block that has PRRS, Seneca A, and PED viruses frozen into this ice block. They toss it into the feed bin and then let that disperse out into a nursery. So 2 rooms, 100 pigs per room, 6 pens per room. In this particular instance of the running the Pipestone Challenge, only the PRRS virus made it into the pigs. So, you can see here we only have that data to compare. So compared to the control group where they got the virus but with no mitigant in the feed, the GML led to about a 67% reduction in positive feed samples for the PRRS virus. We had about a 1/6 as many positive oral swabs for the virus in those pigs. We had none of the pigs in the trial seroconvert, and no clinical signs in any of the pens that were received GML. So, and you can see quite a nice improvement in average daily gain over the negative control pigs, the pigs who had no mitigant in their feed. So we feel very confident that this is a very potent tool for us to use as we try to combat not only these viruses, PRRS and PED, that we've been battling for years, but also with hopefully the one we won't have to deal with for a while, the ASF virus here in the United States anyway. Now, I'll also take this into an in vitro studies for feed mitigation of ASF. Again, work that we did with the National Academy of Science in Armenia, where we took 2 grams of dry feed that had the various treatments blended in, and in this we used a blend of medium chain fatty acids and GML, but we fed it or blended it in at different concentrations from 0.25% up to 2% of the feed. So it was inoculated with the virus and incubated for either 30 minutes or 24 hours. We then added media and spun the feed particles out, leaving the viral particles up in the supernatant. And then we took samples from that to test one of 3 ways, either through virus infectivity, as I've described earlier, or we used quantitative PCR to look for signs of the DNA, viral DNA, in the samples. And then the third approach was to do an ELISA, an enzyme-linked immunosorbent assay, for the predominant protein of the ASF capsid. So P72 is that protein. So we were looking for intact P72 protein through this assay. So we'll start off with the infective virus recovery from the feed. And what you'll see is that the medium chain fatty acids at all of the concentrations we tested were not effective at changing the amount of infective virus. So the virus maintained its infectivity across that range of medium chain fatty acid inclusion. At 1% GML, while not significant, there was a tendency led to about a 0.67 log reduction, I think it was, in infective virus. And then at 2% GML, we saw almost a 1 log reduction in as early as 30 minutes after inoculation. So acting very quickly. And basically those same results were mirrored at 24 hours. Now, as we might suspect, we really didn't do anything that would have altered the amount of viral DNA in feed. We still had infective virus, and clearly we still had DNA in the feed. So we didn't do anything to say degrade the DNA, but basically none of the treatments had any effect on the amount of DNA found in the feed samples. Now we're going to jump to looking a little bit at the structure of this ASF virus, because it's a much more complex virus than some of the others, PED or PRRS, for instance. So this green layer right in here, and I'll try to highlight it here. Maybe that'll do. Yellow on green doesn't work so well. Anyway, that green layer right there is the capsid layer. And that P72 makes up about 92% of that entire capsid around the viral nucleoid. And so P72, if that unravels, basically it will disrupt fusion with host cells and other virus infectivity activities. So as we look for that viral protein, the higher the reading, the more intact P72 we have in the sample. So here we can see a very pronounced effect, as low as 0.5% GML in the feed. We saw a significant decline in the amount of intact P72 protein, and at 1% and 2%, there was further disruption of that P72 protein. So that protein, basically its feet, if you will, are embedded in the inner lipid membrane, and its head is embedded in the outer lipid membrane. So if we've disrupted that outer lipid membrane, we're probably also disrupting the structural stability of the P72 protein. And again, if that's disrupted, then its ability, the virus's ability to infect a host cell is certainly compromised. So I'll start to wrap this up and just tell you that most sources of the medium chain fatty acids and monoglycerides are grass and readily implemented for livestock. They're potent antimicrobials and antivirals with the added benefits of having anti-inflammatory activity and also a very positive attribute of being growth promoting. And I think this is really the first mechanistic data that we've seen of a mitigant demonstrating efficacy against the ASF virus. So we feel like when this paper comes out here in a few weeks, hopefully, that it will have quite an impact on people's understanding of how these various mitigants can work against ASF. And by taking the depth that we do in understanding really the biophysical membrane chemistry and activity, and also the chemistry of potential mitigants, we can really help to target solutions to the specific animal health challenges. And so to wrap this up, I guess I would like to just thank our collaborators for all of their efforts in helping us assemble all this data. And with that, I'll turn it back over to you, Andy.
Thank you, Dr. Elrod, for a great presentation, and thank you, the audience, for joining us for this session of the Global Hog Industry Virtual Conference. If you have questions for Charlie, I would highly encourage you to visit the Natural Biologics booth in our expo hall and to download more resources from them as part of your experience here at the Global Hog Industry Virtual Conference. I'm Andy Vance. Have a great rest of the event.